what we are going to do is we're going to discuss about the motifs, how to analyze the motifs, and then restrictions and how to identify restrictions out in the sequences. Okay. So first, motifs in the sense it's a particular sequence, like three sequences after particular sequences, nucleotides or protein sequences. So you that will be used for specific function. That is a motif. Okay. If you identify that motifs in a particular protein or DNA, then you can say that these are kind of conserved, not conserved. These are kind of having that particular function. Okay. Particular function. This protein kind of having a particular function. One of the examples is glycation, and there is a lot of motifs. And one of the examples we can take is glycation. The for four we can write it now. So import okay. Some bio import step IO. Sorry. Set IO. Then import RE. That is a kind of a model that is used for motor analysis. Okay. Regressive models that is going to used for motor analysis. Then you have to create a variable called set where I'm going to save a one file. Okay, this is the cytochrome MSA that was previously used for multiple sequence alignment. So it's have a different sequences. Okay, same cytochrome C oxidized G from a different species, yeast, rhodobacter, and EcoRI, and then humans. Okay, this is the sample we are going to. This is the DNA we are going to use. Okay, how can we use this? C, I, A, yes. Before all the video, we only use a read. Okay, instead of first, we have used as a read. But here we are using as a first. Why in the sense means the document have a multiple sequence. If it is have only one sequence, then we can go with a read. But it is have multiple sequence. Okay. So what we have to do is we have to first. The first means it can analyze the multiple sequence. Okay, if you put a read instead of first for the multiple sequence, it will give a error. Okay. Then we have to give a name of the document. That is and this is how I saved the document like the cytochrome and message. Okay. Then I have to give a type of file. It is a FASTA agenda that I have to give. This is a FASTA file, so I give it. Okay. Now, what we are going to do is first we are going to print the protein sequence ID because we have a four sequence. So we have to print the protein sequence name ID. Then what we are going to do, we are going to print our motifs. Okay, motifs present or not, which motif is present, and motifs groups, and then the sequence in the position, the motifs position in the gene. Sorry, the protein. Okay. So for that, we are going to create a loop. Why in the sense we are going to create a loop means it's a multiple task, four sequences. It will be, if we doesn't use a loop, then we have to write a sequence for four times. So for that, we can just create a loop, then it will be listed for, we can do the automation. Okay? For, I just create a loop file for loop. Then I'm going to create a one temporary variable called protein. This protein, this protein stable. Okay, now we can start. So I'm going to create a loop that is for loop, and after that I'm going to create a temporary variable called protein. You can create anything, the protein or I or anything, whatever you want. You can create a temporary variable in the set. Okay, so in the set. Okay. Then what happens is every there is a four amino acids and protein sequences present in this document. It will take one sequence, one protein sequence at a time. Okay? So for each protein sequence, what we're going to say is print s dot f apostrophe means saying that whatever inside I'm going to type, this is string. Okay, and also you can put a variable name as well inside the specific bracket. Okay, it's a kind of format to type same variable and then string in the same content, same line. Yes, we are going to protein, then a variable that is protein. The ID. Protein means at the time we take one sequences, that is a protein. That protein it will every time it will be changed. Then ID and from the document it will take that ID name and then it will print. Okay. Then after that what we are going to do is we are going to do a motifs analysis. Okay. Which protein analysis we are going to do now is glycylation. Glycylation, glycylation means it's kind of a protein after post modification. So post modification happen in the Golgi apparatus. It has some protein called N. Uh, any the second question will be anything. Okay, A P A R A R T R S anything. Sorry, A P G C anything. Okay, and any amino acid sequence will be there. Okay. Then after that it will be SRT. Okay, in that specific. So it will be this is the motif structure. Okay, so we are going to identify this structure. Okay? How can we identify in this instance? Again, we are going to create a loop because every time it's a 4 sequences. So each time we have to do that. Okay, so for I'm creating the variable I. Okay, then I'm input in that means RE means I'm just calling the tool here RE and then inside that again I'm just calling another function that is called find before. Okay, then I'm going to write this R N dot S S T. Okay, what it means R means it's a kind of formatting. Okay, it will format your the sequences and sequences that is you given here. N means that N amino acids you're going to identify, and dot means the second amino acid should be anything. Okay, it's RNA, anything it should be identified. Okay, then third amino sequence should be either S or T. Okay, this is how we're gonna write it. Find bitter means what its role here is every time whenever it identifies the motifs, this motif inside a sequence, it will create a group. Okay, it will create a group in the backend that we cannot see. It will create a group. In the group, it will store the name of the motifs. If it is NAT, it will store as NAT, and this is the position. And if it is a N AS, then it is a NAS, and it will show a position number as well, which is it is in the 321 or 320. Okay. Then what we have to do is str. Str means string. For the RE function in the RE tool need a perfect string. Okay? So before that, what we are going to do, we have to change the protein dot seq. And the protein dot seq, we have to change it into string. Then we have to print it. Print. It's notice. Then match dot group. Group means it already saved the motive name inside the group. Okay, that's why we have to do it like this. And portion number we have to put for that. For that we have to write a portion and create a variable called match dot start. Why match dot start in the sense like we have a 3 nucleotide now. If you analyze the sequences, so you identify NAT. So start means the N where it start the sequences. And we have to give +1. Why +1? In the indexes of nucleotide, and the indexes happens in the Biopython start from 0, not from 1. So whatever the nucleotide present, it's always one nucleotide on protein amino acids are there. No? So that's why we try to add +1. Here we have to put. Now I just run this. Some poetic things can be put here. New line. Slash n means you're saying that it's a create a new line. See, it depends a protein ID. It's a yeast one, and motive is NQT. It's present. So at the position of 20 seconds. See in the rhodobacter sequences. This is the rhodobacter, and position motive is NLT and 237. And in the EcoRI, it having a two motives, NUT and VP. And version is 42 to 79. In the human species, there is no motives present, so it's just giving us a blue. So this is how you have to analyze motives. Not only NSMAT, there is a too many, like whenever you go with a specific work, you have to identify specific motives. At the format, you can create, you have to change this one to identify the motives, specific motives you want. That's it about the motives. Now we are going to identify the restriction site, how to identify the restriction site with using Biocode. That how can we do this? We can input uh, bio.set bio.set input set. Okay. Then again from bio import SeqRI SeqRI. Okay. Then what we are going to import is if you want to identify specific uh, restriction site, okay, then what you can do is from bio dot restriction info. Yeah, if you want EcoRI, okay, EcoRI, then you can just input like this, and you have to create a DNA. Okay. DNA, that is, I have on sequences by, we can use this sequences, okay, TB53, mouse, that is from mouse that we can use. DNA is equal to SeqRI from read. Why here we use read means it's have only one sequences. Okay? Only one sequences. So if I have multiple sequences, then I can go with the times. Something like that. Here I have to give a file that is 53 maps. That's fast. Then we have to give a fast file type. That is a fast file. Okay. Then I have to put .seq. Why we are saying this one is we are only telling the Python only take sequence apart from the sequence ID and name, everything leave. Okay, we are just saying that only download and only take the sequences. Okay, again stay unstable. Okay, so again saying that set file dot read and TB53 mouse. This is the file name that I was like put it here. This is the file I'm going to use. Sorry, this is the file I'm going to use. So that is why I'm putting here. And I put the file type FASTA and put a .seq. Why means like you are saying the Python, you are saying Python to only take sequence apart from the sequence ID and sequence description. Everything leave alone, just taking the sequence only document. Okay. Then what we are going to do is you are going to call the EcoRI. Okay, this is this function. This function contains the uh, restriction nucleotide, okay? Which restriction site nucleotides. It's have a GA, GC, no? That nucleotide present inside the function. So you just directly use EcoRI. EcoRI dot search DNA. Um, you will be able to search restriction site. So this will not print. See, it doesn't have any restriction site for EcoRI. Okay, so I'm just changing the another answer. I mean another restriction site. That is HindIII. Here also I'm just going to change the name of the enzyme. I mean the restriction site HindIII. Okay, so again I'm just running. Again it says doesn't have any HindIII restriction site here. So what I'm going to do is I'm just going to not only just go with the one restriction site, I'm going with the all the restriction sites. Okay, going for analyzing for this particular sequence. Okay, so I'm going to identify uh, is there a restriction site any other thing present. So for that, I'm just putting all enzyme, all the restriction enzyme. Here also I'm putting inside. Like you can create a loop for us. Start I in the all enzymes. Okay. Because all enzymes having no, like all enzymes having the with hundreds of restriction enzymes. Okay. So in that you are just taking one enzyme, sorry, one restriction enzyme at a time to identify the specific, identify the that specific site present in our sequence or not. Okay. That's why we are going to create a loop. For I in the all enzymes. Okay. So I'm creating variable called sites. Okay, sites in the sense you are creating I dot means the variable stores the restriction enzyme name. Okay, then you're going to give it that and then search in TM. Then again you're going to create a one that is if sites presence. Okay? If sites are there, then print uh, I the restriction enzyme then sites. Whereas the sites are present. Okay? Okay, now this make it full screen. So it's having these others, RALI, and that is having the this sequence is present in this system, and ABSI, this restriction psi will be present in the sequences. That is 33. These are the different different places it has. Okay? So whenever you are using this restriction enzyme, so it will be cut in the specific particular state, all the spaces. And MSPGL, FSPGL, all the things. Okay? So you can identify restriction site using the Biopython. It's very simple. Tomorrow we can, this is what I want to discuss with you all today. And tomorrow we can discuss about the pipelines, how to create a pipelines with, for the any process. Okay.